Okay, and something has gone ding. Right. Two phyla, one of which is exclusively parasitic but not very well known. The other one of which is very well known but doesn't have many parasites, but that I mentioned because there's a distinct possibility you could find it around here. The first being the Mesozoa, which not a lot of people have heard of because it lives exclusively in cephalopod kidneys. So there aren't very many of them in Arkansas because we don't have many native squid. The other's the mollusca, that is clams, snails, squid, slugs, a number of other types that are not as familiar, like chitons and scaphopods and monoplacophorans and such. But, you know, everybody's heard of clams and snails, squid, octopus, things like that. There's not very many parasites in that group, but there's one that's quite interesting, and one that you can encounter around here. One that we probably, I don't think we saw it in last week's lab, but it's possible that the Hufflepuffs might have better luck. Anyway, first with the Mesozoa, they're so simplified that we don't really know where they belong. They're possibly platyzoans. Remember, that's the group with flatworms and acanthocephalans, but this is not entirely clear. The anatomy is so simplified that it's hard to tell. And last I looked, the DNA analysis was not really conclusive as of figuring out where these guys belong on the tree of life. And it's an open question as to whether they are secondarily simplified or have always been really simple. We've seen this before, that parasitic taxa often strip down a lot of the complexity in their free-living relatives. We really saw that, if you remember, the Myxozoa, those critters that were thought to be protists for a long time, but are basically jellyfish that have been completely minimalized. And you get this in the Mesozoa as well. It's possible that these are flatworms that are so simplified it's no longer obvious what they are, but nobody really knows. There's two classes, and they're not even necessarily closely related to each other. Dicyemids are the parasites of squid kidneys, and orthonectids parasitize a lot more critters and have a really bizarre life cycle. So that's what a dicyemid looks like. There is a single long cell running from head to tail called an axial cell. And the axial cell is surrounded by a jacket of anywhere from 9 to 41 cells. Sorry, somebody just went ding. There we go. Good morning. And in all members of a species, that number of outer cells is the same. You can identify species in part basically counting the number of external cells that there are. The fancy name, by the way, for a case where an organism or a structure always has the same number of cells is called eutely. So dicyemids are eutelic. You see this in other critters as well. Roundworms in particular are often eutelic. It's made one particular roundworm called Caenorhabditis elegans a very popular model organism in developmental biology, because the path of development from fertilized egg to, I think, 939 adult cells is always the same. That's not directly concerned with parasitology, but it's a case where eutely actually has a practical application in a very important biological field. Anyway, the cells at the anterior of a dicyemid form a cap-like structure. It's called the calot, not the culotte. That means breeches. The calot is what's at the top. Here you've got 8 different calots from 8 different species. Species are identified by looking at the shape and size of the calot, and the calot is what attaches to folds and pockets inside the cephalopod kidney, renal appendage. So there they are. The life cycle's weird. Embryos develop from extra nuclei inside the axial cell. The axial cell has multiple nuclei, and during the asexual stage, those extra nuclei just divide into embryos, which get out somehow, I'm not even sure how, and develop into a new round of adults. So we've seen this before, this alternation between sexual and asexual stages. Asexual reproduction produces embryos that look like miniature parents. They're called vermiform embryos. Vermiform means shaped like a worm. When population density is high, they start reproducing sexually. I'm not sure we know the details of how, but they begin to produce a different type of embryo called infusoriform, which basically means it's shaped like a ciliated protist. You can see it's differently shaped and covered with much longer cilia. Those somehow leave the host and find new hosts, and we don't know how they do it. It strikes me, I just thought of this, in a lot of cephalopods, a lot of them are, what's the word, semelparous, meaning they reproduce all at once and then lay their eggs and then all die, meaning that there may not be any contact between generations. The babies may be born completely as orphans. That suggests to me that the infusoriform might have to live on its own for some time. If you have these, if you have these breaks, if you never have older ones living together with younger ones, that might say something about the life cycle of the infusoriform, but I don't really know what. There's not a lot of people that have deeply studied these. One of the few people that did, in fact, the discoverer or the namer of the phylum Mesozoa was a guy named Eduard van Beneden, who did a lot of parasitology in the later 19th century. And Dicyaemids, thanks to him, scored an important assist in the history of biology. Van Beneden did studies on a number of parasite groups, a lot of them on nematodes, roundworms, but he was looking at Mesozoans and noticed that fertilized eggs, the ones that would give rise to those infusoriform larvae, the sexual generation, the fertilized eggs in the axial cells did something weird with their nuclei during the first cell divisions. And this is his diagram of early cell divisions in Mesozoans. And they might look a little different from what you remember, but you can clearly see that he's drawing mitosis. Figure 13, the one at the upper right, is pretty clearly chromosomes lined up on a metaphase plate. So that's metaphase. And then by 11, you can see chromosomes on the metaphase plate, a full set of spindle fibers, and a couple of little round things at the top and the bottom of the meiotic spindle. This is diagram 11 at the lower left. You can clearly see that's a mitotic spindle and centrosomes. I'm hoping you still remember that from Biology 1440. Van Beneden was not the first. The first was this German guy named Schneider, I think, who was looking at salamander tadpole development. But Van Beneden was one of the first to describe mitosis and the first to see centrosomes. So every so often, studying something really weird that nobody knows anything about and nobody would think might be interesting can lead to some pretty big payoffs. This is a pretty big development, important for understanding cell division, and then ultimately that ties into genetics and all of that. He'd go on to, this is why there's a statue raised to him, by the way. He'd make much better observations of cell divisions when he started studying parasitic roundworms in the genus Ascaris. This is one that you can have so many of them in your gut that they can block the gut, which will kill you if you don't get it surgically treated. This is the human intestinal roundworm. There's actually a close relative that infects pigs. And studying these in 1883, he would describe what we now call meiosis. And he is credited as being the first person to properly describe meiosis. So he's a co-discoverer of mitosis and a discoverer of meiosis, and it all begins with studies of obscure parasites living in squid kidneys. So if there's a lesson there, it's that every now and then you can get big payoffs from studying the weird and obscure things that you don't think anybody would care about. Which reminds me, years ago when I took microbiology, there was a cartoon in my lab manual. There was a guy looking down a microscope, and another person in a lab coat was saying, "There are people dying out there, and all you can do is indulge your curiosity looking at mold, Dr. Fleming." The joke being that Alexander Fleming discovered penicillin. Right, anyway. Second class of Mesozoa is even less well known, although they parasitize a variety of marine invertebrates. I know there's some in echinoderms, for example. The adults consist of a layer of ciliated cells. This is what they look like from the outside, surrounding a mass of sex cells, not a single axial cell like in the dicyemids, but a mass of sex cells. That's a male at the left and a female at the right. I won't say much about the life cycle. We know that male sperm burrow into females, fertilize their eggs, give rise to ciliated larvae that swim around until they find new hosts. The larvae form syncytia, which bud off new males and females. And that's all I know about them. So they're there. Riffing on what I was talking about just earlier, maybe there's something really interesting about basic biology that we could learn from studying orthonectids. Hey, maybe one of you could go on to be the world expert on orthonectid Mesozoans. It's not like it's a very crowded field. Okay, maybe you shouldn't take too much career advice from me, but, don't ever feel like we've solved all the problems. We haven't, and there's lots of interesting things that researchers have never really looked at in proper detail. We're not going to run out of interesting things anytime soon. Okay, right. So here we're coming back to animal classification. I've finished talking about platyzoa. We've said all I'm going to about flatworms and acanthocephalans. Maybe Mesozoans go in here, but nobody knows. We've talked about all the protists I'm going to. When we talked about Cnidaria, those are mostly free-living, but we've talked about the mixozoans, animals that are bilaterally symmetrical are divided into protostomes and deuterostomes, and I've said all I'm going to about deuterostomes because there's not many parasitic deuterostomes. Just lampreys and eels living in shark hearts and things like that. Protostomia, we've talked about platyzoa. The ecdysozoa is the second group that's got lots of parasites because that includes nematodes or roundworms and arthropods, which have a lot of parasitic members. everybody from fleas, ticks, and lice to those Caribbean sand fleas that burrow into your big toe, kind of unpleasant, to the mites that cause mange and scabies, to parasitic wasps that do such horrible things to caterpillars. Today, I'll start on the Trochozoa. That's not a major group known for lots of parasites, but it includes segmented worms, which itself includes the leeches. We ought to talk about leeches, this being, a taxon you can readily find in Arkansas if you go wading in your bare feet enough. And it also includes the mollusks. Mollusks and segmented worms are in a large group called the Trochozoa. And what unites the Trochozoa is that, at least ancestrally, they have a trochophore larval stage. And on the left, there's a picture of what one of these looks like. It's basically shaped like two rounded cones stuck blunt end to blunt end. There's a gut. You can see the stomach pretty well at the upper left of the picture on the left. And on the right, there's a much larger stomach shown in the diagram. The mouth is kind of on the side. The anus is below in these pictures. There's twin bands of cilia around the widest parts, around the equator, if you like, and then tufts of cilia at both of the sharp ends, at both of the poles. This would have been present in the common ancestor of mollusks and segmented worms. Certainly marine species of both go through a trochophore larval stage. You can hardly tell a marine segmented worm's trochophore apart from a marine snail's trochophore. They look so similar. Now, some species of both have lost this stage. Earthworms don't have a trochophore larva because they don't reproduce in water. Reproducing on land means that they've highly modified the way that they develop. They develop directly, not through a larval stage, but just straight into a miniature adult. Leeches, I think, do the same. They don't go through a trochophore stage, and then some mollusks don't. Squid, for example, don't. But we think it was present in the common ancestor of annelids, mollusks, and a couple of other phyla that we don't need to get into for this class because they're not parasites. Okay, so mollusks. They're a diverse group, and you might not be aware that everything that we classify as mollusks really do belong. There's no single feature that every mollusk has, but most members have most of the following. Most will have a shell. Some don't, of course. Most octopus have no shell. Some actually do. The familiar ones don't. Slugs, of course, don't. There's a few others that don't. But most have a shell, and the shell's made of calcium carbonate, CaCO3, deposited in an organic matrix that's made up of specialized proteins, deposited under very careful chemical control. There's a lot of energy required to make a mollusk shell because those proteins are secreted in such a way that they control very carefully exactly where the calcium carbonate goes. The shell is secreted by a flap of tissue called a mantle. You'll see a very nice mantle right at the end of this. There's a cavity between the mantle and the main body that's called the mantle cavity, and that usually contains feather-like structures called ctenidia, aka the gills. The gills are, of course, used for respiration. In clams, they're used for feeding as well. They are the primary filter feeding organs. In snails and squid, they're not quite as big, but they're still definitely present. Most mollusks have inside the mouth a ribbon-like feeding organ that's kind of like a very flexible file with lots of little teeth made out of chitin. That's called the radula. There can be other things in the mouth. Squid, for example, and octopus have a beak. Superficially looks like a parrot's beak, but they can take bites out of things and sometimes inject venom. But they've also got a radula inside the mouth that they can rasp back and forth over whatever they're feeding on. You can see this if you've ever watched a big snail crawling up the surface of some aquarium glass, for example. You'll see the snail. It's kind of hard to describe, but it almost looks like it's licking the glass. That's the action of the radula. Being dragged kind of back and forth over the glass in a motion that's a little bit like you using a towel to dry your back, kind of a back and forth rocking motion. And the fourth feature that most mollusks have is a muscular part called the foot that's primarily used for locomotion. In a snail, the foot is what the snail is crawling on. It's, you know, the slimy bit that sticks out of the shell and that pushes the snail forward very, very slowly. In clams, the foot is narrow and used for burrowing. In squid, the foot has evolved into the tentacles. Other groups have different types of foot, but most mollusks have most or all of these basic features. Although you can find exceptions. Clams don't have a radula. Slugs don't have a shell. This oddball group of tropical mollusks that haven't been well studied, called the aplacophorans, don't have a foot, and so on. But by and large, this works for the mollusca. Vast majority of them are perfectly free living. Snails, for example, most of the ones that you'll ever see just, you know, crawl around and feed on algae or what have you, or smaller critters. There are some that are predators. There's a few snails that are parasites. What you're looking at here is a sea cucumber. That is the kind of pinkish, bumpy thing up and to the left of this picture. And the white things that you see in the center are a couple of small gastropods, maybe a couple of millimeters long, in the genus Melanella, that are hanging onto a sea cucumber and piercing its skin with their probosces and slurping up its bodily fluids. When I was in 6th grade, we took a long vacation to Hawaii because we actually had family at the time living there. And so we stayed with them. And of course, we went to the beach a whole lot. And I can remember going to a beach on the island of Maui and finding these giant, you know, foot-long, black, lumpy sea cucumbers and picking them up and actually seeing these little white snails just sitting there and slurping out their body fluids, being ectoparasites. I also made the discovery that when startled, sea cucumbers could squirt a jet of water out of their anuses. So naturally, I made good use of this discovery and started picking up sea cucumbers and squirting my sister with them. And we had kind of a water gun battle using the water squirted out of sea cucumber anuses. So yeah, we're both pretty much scarred for life. But yeah, I've actually seen these before. You know, it was kind of a formative experience for the 11-year-old me. I wish you similar experiences, except maybe without the squirting your sister with water out of a sea cucumber's butt. That's kind of weird. Anyway, I digress. There are others that can actually penetrate into the tissues. That's a starfish in genus Lynchia with another snail, genus Stylophor, that's actually made a cyst inside the arm tissues of this starfish, and the cyst has been dissected out. So that little pink coily thing in there is a snail. Some members of this family have gotten so good at endoparasitism that they've lost the shell and completely lost the gut. So this is a eulimid. This is in the same family as the two that we've seen before, but it's completely ditched its shell. It's completely lost its gut. It absorbs nutrients right through the body wall. It lives in the intestines of sea cucumbers, and it's basically a snail that thinks it's a tapeworm because it's lost all of those molluscan features. You probably can't tell that it is, evolutionarily speaking, a modified snail. You can tell by looking at the larvae, because the larvae are your typical trochophores. Tapeworms never go through anything that looks like a trochophore. They might go through that swimming coracidium stage, but they never are trochophores. So looking at the larvae is how you can tell that Enterozinus isn't a weird tapeworm, it's a weird mollusk. So just another great example of how parasitism can be accompanied by extreme anatomical streamlining. But the most awesome molluscan parasites, and the ones that you might actually have a chance of seeing if you poke around in streams in Arkansas, are actually not parasitic as adults, but they have a parasitic stage when they're juveniles. This is the freshwater mussels in the family Unionidae. And people don't always realize this, but the southeastern United States is a surprisingly biodiverse area. It's a biodiversity hotspot. Not quite perhaps on par with the Amazon rainforest, but there's a whole lot of unique species in the southeastern United States, including nearly 300 species of freshwater mussel. Now, they have these in other states as well, but diversity is concentrated in the southeast quarter of the United States. Unfortunately, over 70% of our freshwater mussels are either extinct or thought to be extinct or endangered, threatened, or of concern. And the usual suspects, of course, are things like dam building a dam on a stream. Back of the dam, sediment tends to build up, and water, of course, slows down because you make a lake or a reservoir. A lot of these mussels need very clean and very fast-moving water and can live perfectly well in a stream, but they can't make it in a lake-type environment. And, of course, you've got pollution and, hog farm waste dumped into rivers and all sorts of things like that, the usual. But a reason that doesn't apply to other species is that freshwater mussels were very intensely harvested in the 19th and early 20th centuries for making buttons. The center of the button industry in the U.S. Was actually this little town on the Mississippi River called Muscadine, Iowa. One of the settlers was a German immigrant who'd learned how to make buttons, and he set up a factory at Muscatine. Other factories moved in, and pearl buttons used to be cranked out by the millions between about 1880 and I think 1940 was about when plastic finally started replacing a natural shell. But yeah, they used to harvest pearl buttons all over the United States. It actually used to be a thing that Arkansas farmers would take a little time off in the summer between planting and harvest, and they might actually go camp down by the creek, and of course, have a little bit of recreation and swimming and camping and things like that. But they'd also collect freshwater mussels and save the shells and sell them to dealers that would go on and sell them to the pearl button factories. That's part of the reason why we have 2 endangered species within the state of Arkansas. The Arkansas fat mucket, with a lovely name, shown on the left, and the Ouachita rock pocketbook mussel, shown here on the right. They're both restricted to rivers in southwestern Arkansas, and I'm not sure if either one was heavily harvested for pearl buttons, but they've been affected by building dams, pollution, silt buildup, agricultural runoff, you know, all of the usual things that tend to make species endangered. And I've often thought that if I ever have $50 million that I don't have a better use for, I would donate it to the UCA athletic department on condition that they change the mascot to the Fat Muckets, just because I'd like to watch the athletic director's head explode. But, you know, it's a uniquely Arkansas thing. There's lots of teams named the Bears, but yeah, nobody's named the Fat Muckets. You know, we could do instead of the tomahawk chop, we do the clam clamp or something like that. It'd be great. Here's one that used to be restricted to the Little Red River north of us. Flood control projects wiped it out of the north and the south fork of the Little Red. It's now only found in the Middle Fork. And then when the Middle Fork was dammed to create Greer's Ferry Lake, that took out most of the rest of its habitat. And it's restricted today to a 9-mile stretch of the Middle Fork of the Little Red River upstream from the town of Shirley, which maybe some of you have seen. I have been to Shirley a couple of times. You may know it. So where's the parasitology here? It's great that we have all of these clams. Unionids go through a parasitic larval stage. Instead of releasing those trochophore larvae, the fertilized eggs develop brooded on the mother. The gills are modified to produce a pouch. These are called marsupial gills. Yes, just like marsupial in mammals. These have a pouch. And the fertilized eggs develop into a little larval stage called a glochidium. So there's a glochidium at the upper left. It's basically a miniature clam, but you'll notice that the clam has rather sharp pointy bits. And that's because when mommy clam squeezes her marsupial gills and releases the glochidia, they snap their way through the water, kind of like little aquatic Pac-Man, until they land on a fish's gills. And they'll latch onto those gills and feed on the fish's blood, although not enough to really do the fish much harm, and grow on the fish's gills until they eventually drop off and mature into adults. So there on the lower right, you can see what these glochidia look like latched onto a fish's gill filaments. This rarely does serious harm to the fish, but it does mean, of course, that the clams can't reproduce unless there are fish in the same stream. So anything that affects the fish can also affect the clams. And here's the really cool thing. All clams have the sheet of tissue called the mantle that basically lines the interior of the shell, and the edge of the mantle secretes new shell. That's how the clam gets bigger. But in the Uniona genus Lampsilus, the posterior mantle is enlarged. So there's a flap kind of sticking out of the posterior end, the end that sticks up and out of the bottom. And in some species, all you have is this flap. But in other species, the flap is actually modified. Its shape has evolved to resemble a small fish. And the colors have evolved in some species to mimic a fish. In this one, you can actually see there is an eye spot over on the left-hand side of that flap. and there's little extensions off the flap that looks like fish fins. So this mussel has a flap on it that looks like a little fish. When a larger fish, like a bass, swims over to investigate the lure and say, hey, can I eat that? If the bass, for example, tries to take a bite out of the lure, it gets a face full of glochidia, because that's right at the moment when the clam will just fire off a bunch of glochidia and hit the bass right in the mouth, and the bass's gills get parasitized by the glochidia until they eventually drop off. It's an awesome case of what in evolutionary biology we call exaptation, taking a structure that's already there and modifying it. And it's fully explainable by your old friend and mine, natural selection. Lampsilus that has a flap that looks more fishy will tend to leave more babies than Lampsilus that doesn't have a flap that looks more fishy. The more fishy that flap looks, the more offspring you leave. Wash, rinse, and repeat over many generations. And you've got Lampsilus with these surprisingly lifelike lures that even look like they're swimming when they wriggle. So it's not all species are equally fish-like. There are some where the flap doesn't look like anything special, but certainly some species of Lampsilus have taken this to an amazing extreme. And I think it's really cool that, we are not the only species that uses artificial lures to trap fish. Okay, right. So I'm going to go ahead and stop recording now.